Welcome to the Biology Brief channel. Today we are starting a MCQ discussion session regarding the chapter Molecular Base of Inheritance. This contains 3 parts. Today, in first part, 30 MCQs are discussed. The questions from nucleic acid, structure of DNA, genetic material, and DNA replication. We are going to first question. Yes, first question. Adenine plus ribose plus phosphate form. Here, adenine plus ribose, adenosine. It is a nucleoside. Then we have to add one phosphate with it. Then it becomes adenosine monophosphate or AMP. Another name for AMP is adenylate acid, and this is our answer to this question. So adenine plus ribose plus phosphate form adenylate acid. Other options are wrong. Yes, let us go to next question. Select the mismatched pair from the options. There are 4 options given. If you see the datas, these are the nitrogen base, number of nitrogen bases in 4 organisms. If we look at, you may see that the figures are just like as given in the textbook. But if you carefully examine the options, first one 5x174 is a bacteriophage which contains single stranded DNA. Therefore, in that DNA there are nitrogen bases or nucleotides, not base pairs. Here given 5x174 bacteriophage contains 5386 base pairs. This is a wrong statement. So, bases. 5x174 bacteriophage contains 5386 bases. So, the first one is a mismatched pair. Others are correct matching pairs. Yes, moving to third question. Which one of the following is not a component of nucleotide? Not a component of nucleotide. A nucleotide consists of nitrogen base, pentose sugar, and phosphate group. The first 3 options are components of nucleotide. Therefore, our answer is the 4th choice, that is disulfide bond. Disulfide bond is present in polypeptide chains. Here, when 2 amino acids with sulfur come closer, the 2 amino acids will be linked with forming a disulfide bond. So our answer to this question is option 4. Moving to next question, the number of base pairs in haploid content of human DNA. You may confused with options 2 and 4. You all know that the diploid number, in diploid number, the DNA content in human being is 6.6 into 10 raised to 9 base pairs. When it comes to haploid number, you have to take the half of this figure. That means 3.3 into 10 raised to 9 base pairs. So, 4th choice is our answer to this question. Moving to next question, which of the following is an incorrect statement? You have to select incorrect statement from the options. First one, cytosine is common for both DNA and RNA. It is a correct statement. In addition to cytosine, adenine, guanine are common for both DNA and RNA. A dinucleotide contains 3'5' phosphodiester linkage. A dinucleotide, sugar, nitrogen base, then sugar, nitrogen base. These 2 nucleosides are connected by phosphate group. Here 5', here 3', and here the phosphate group makes 2 ester bonds with these 2 nucleosides. So, the statement is also a correct one. A dinucleotide contains 3', 5' phosphodiester linkage. The 2 strands of DNA have parallel polarity. If we examine the statement, we know that there is no parallel polarity. The polarity is anti-parallel. That means one strand. One strand runs in 5', 3' direction. Then another one 3 prime 5 prime. The two strands are anti-parallel. 3 prime to 5 prime and 5 prime to 3 prime direction. Two strands. So the third option will be our answer, which is a wrong statement. The nitrogenous base in two strands of double-stranded DNA are paired through hydrogen bonding. It's a correct statement. Here, adenine pairs with thymine with the help of 2 hydrogen bonds. Guanine pairs with the cytosine with the help of 3 hydrogen bonds. The fourth statement is also correct. For this question, third choice will be our answer. Sixth question, in a 3.2 kilo base pair long piece of DNA, 820 adenine bases were found. What would be the number of cytosine bases in it? Here, the length of DNA is given as DNA is equal to 3.2 kilo base pair. That means 3200 base pairs are totally in it. Here, A plus T, A plus adenine and thymine are more or less equal. 820 plus 820 equal to 1640. Here guanosine and cytosine are also equal. Their number will be 3200 minus 1640, that is 1580. The number of cytosine is asked here. So the number of cytosine is 1580 divided by 2, that is 780. You can easily calculate and find out the correct answer. 780. Option 1 is given as 780, and that is our correct answer. Then move to next question. Nitrogenous bases are components of monomers of nucleic acids. How many nitrogenous bases are common in DNA and nucleotides? Here, nitrogen base, DNA, RNA, adenine, guanine, cytosine, thymine, uracil. Adenine present in DNA and also in RNA. Guanine, cytosine. First three nitrogen bases are present both in DNA and RNA. What about thymine? Thymine is present in DNA but absent in RNA. Uracil is absent in DNA but present in RNA. Here the question is, how many nitrogen bases are common in DNA and RNA nucleotides? The first three, adenine, guanine, cytosine, are common in DNA and RNA. So 3 will be our answer to this question. It's an easy question. Moving to next question. 5 statements are given. Clearly read the statements and find out which is the correct option from the following. Nitrogenous bases are linked to the ribose sugar by N-glycosidic linkage. It's correct. Sugar. It is correct statement. Sugar, nitrogen base. Then here 3 prime, 5 prime. Here also sugar, nitrogen base, 5 prime. Here phosphate group is attached to this 5 prime. Here the question is the linkage between ribose sugar and nitrogen base. Here this is glycosidic linkage. The first statement is correct. Second one, phosphate group is linked to 5 prime OH of the nucleotide through phosphodiester linkage, 5 prime. The phosphate group attaches to this 5 prime with the help of a single ester bond. Phosphoester bond, not phosphodiester linkage. So this statement is a wrong one. 2 nucleosides are linked through 3 prime 5 prime N-glycosidic linkage, not N-glycosidic. Here 2 nucleosides linked with phosphate group with the help of two ester bonds, one with 5 prime and the other with 3 prime. Here 2 nucleosides are linked through 3 prime 5 prime phosphodiester linkage, not a glycosidic linkage. This statement is also a wrong one. Negatively charged DNA is wrapped around a positively charged histone octamers to form nucleosomes. You all know that DNA or nucleic acid is negatively charged. Here it is wrapped around histone protein, histone octamers, histone protein. Histone protein contains mainly basic amino acids such as lysine, arginine. So the statement 4 is a correct one. The chromatin that is stained dark is euchromatin. The statement is a wrong one. The chromatin that is stained light is euchromatin and it is transcriptionally active also. So the wrong statements are the second, third and fifth. So the fourth choice will be our answer to this question. We are moving to the ninth question. Which of the following is not a feature of double helix structure of DNA? We know that the double helix structure of DNA was proposed by Watson and Crick. And they proposed a model of DNA called BDNA. Here, what about the coiling of BDNA? The BDNA, the two strands are called right-handed and having a pitch of 3.4 nanometer. The fourth option is correct one. The uniform distance between two strands is due to the specific base pairing. If at one strand there is a two-ringed purine nitrogen base, the other strand will contain a single-ringed pyrimidine nitrogen base. So every time the two ringed nitrogen base is paired with one ringed nitrogen base. So the distance between the two strands is maintained uniform, and the option third is a correct one. The two strands of DNA have anti-parallel polarity. Is correct. When one strand runs in 3' 5' direction, the other strand will be in the 5' 3' direction, and the second option is also a correct one. The backbone of DNA is sugar phosphate. Up to this is the statement correct. Then read the remaining portion. Nitrogenous bases project outside. The nitrogenous bases are projecting towards inside as per the double helix structure of DNA. Answer to this question will be the first choice and you can choose it. Moving to next question, assertion reason type question. Given below are two statements, one is assertion, the other is reason. Examine then choose the correct option given below. Assertion. The opposite strands of DNA chains are not identical. Correct one. But complementary to each other. Let us see. One strand is 3', 5', the other one is 5', 3'. Here nitrogen bases A, T, G, C. The nitrogen bases in the other strand will be T, A, C, G due to complementary pairing. If we examine, these two strands are complementary to each other, correct. But if we examine the nitrogen base sequence, here it is ATGC, here it is TACG. The two strands are not identical with their nitrogen bases. So assertion is a correct one. What about reason? Specific base pairing occurs between a purine lying opposite to pyrimidine. Here there is specific base pairing between purine and pyrimidine. That is why the opposite strands of DNA shows complementarity to each other. So our answer to this question is both assertion and reason are true and R is the correct explanation of A. You go with the first option. Moving to next question. If the length of E. coli DNA is 1.36 millimeter, calculate the number of base pairs in E. coli. Assume the distance between consecutive base pairs is 0.34 into 10 raised to minus 9 meter. Yes, there is no need of calculation in this case. You don't waste time. It is directly given in the textbook that as the number of base pairs in E. coli DNA is 4.6 into 10 raised to 6, and you can directly choose the second choice as the answer to this question. Moving to 12th question, it is also an assertion reason type. What about assertion? In E. coli, DNA is scattered throughout the cell. It is a wrong statement. In in E. coli, the DNA is organized and the DNA associated with positively charged proteins and arranged at the center as nucleoid. In E. coli, there is no defined nucleus. The reason is correct one. Correct, because E. coli is a prokaryotic organism. It is prokaryotic and no defined nucleus. There is only nucleoid. In that nucleoid, the negatively charged DNA are associated with the positively charged proteins. So our answer to this question is assertion is wrong. Assertion is false, but reason is correct. Fourth choice will be our answer to this question. Moving to next question. Identify the basic amino acids from the following. We know there are 20 standard amino acids. 20 standard amino acids which make a polypeptide chain. Here, 3 are basic amino acids. 2 are acidic amino acids and the remaining 15 are neutral amino acids. The basic amino acids are lysine, arginine and histidine. The 2 acidic amino acids are glutamic acid and aspartic acid. Remaining are neutral amino acids. Here from this option, you can choose lysine, arginine are basic amino acids. So our answer to this question will be both A and C. What about valine? Valine comes in the 15 group. Valine is a neutral amino acid. Okay. Next question. Again assertion reason. What about assertion? The length of typical human DNA is approximately 2.2 meters. The data is already given in the textbook. We know that. What is a typical human DNA length? That is 2.2 meters. The assertion is correct one. What about reason? The total number of base pairs in human DNA is 6.6 into 10 raised to 9. The reason is also correct, but there is no relationship between assertion and reason. So our answer is both A and R are correct, but R is not correct. Explanation of A. Choice 2 will be answer to this question. Moving to next one. The structure seen as beads on string structure when viewed under electron microscope is here histone octamer. We know that histone octamer is the core around which the DNA is wrapped. Here there are 8 components. 8 globulin proteins are there in the histone octamer. The nuclein. Nuclein is the acidic substance discovered by Frederick Miescher from pus cells. So it is equivalent to DNA and it is not the answer. Chromatin fiber. The chromatin threads are packed to form chromatin fiber. So the fourth option, nucleosomes. If we examine beads on string, like this, the beads are nucleosomes. This is the chromatin thread. Nucleosomes and chromatin thread makes the beads on string structure. So our answer to this question will be nucleosomes in chromatin. Moving to next question. It is a statement type question. Loosely packed and lightly stained regions of chromatin are called heterochromatin. Wrong one. Here loosely packed, lightly stained regions of chromatin which are transcriptionally active are called euchromatin. Then densely packed and darkly stained inactive, transcriptionally inactive chromatin are called not euchromatin, they are called heterochromatin. The terms are misplaced in both statements. So the two statements are false one. So our answer will be option 2. Both statements 1 and 2 are false. We have to select second choice as answer to this question. Then moving to next one. Find out the mismatched pair from the following. Let us start from the bottom. What about nucleoid? We know that nucleoid is the nuclear center present in prokaryotic cells such as E. coli. Negatively charged DNA with positively charged proteins. This is the type of association. So fourth option is correctly matched one. Then heterochromatin. What are heterochromatin? Densely packed and darkly stained transcriptionally inactive chromatin is called heterochromatin. It is also a correct match. Nucleosomes in chromatin we already saw. Beads on string like structure nucleus, it is also correct matching. Histone octamer. spherical protein containing H1, H2, H3 and H4. Analyze carefully. Here, H1 is not present in histone octamer. It is present outside the histone octamer separately, attached with the linker DNA. In histone octamer, the spherical proteins are H2A, H2B, H3 and H4. So the first one is a mismatched pair. You have to select choice 1 as answer to this question. Moving to next question. The association of histone H1 with a nucleosome indicates when DNA associates with histone octamer or his H1 protein, it shows condensation or packaging of DNA to form chromatin and consequently to chromosome. So it is part of the association is part of packaging or condensation. So it is a straight question. We have to move to the third option, that is condensation of DNA in the chromatin fiber will be answer to our question. Transcription is occurring, DNA replication, unwinding. In such cases, there is no association, there is dissociation of histone proteins from DNA. So they are not the answer. Our answer is third option. Let us move to next question. A series of experiments were conducted by Frederick Griffith in 1928 on transforming principle with, we know that, in search of genetic material. The first remarkable experiment was done by Frederick Griffith. He discovered the transforming principle, but he was not able to identify the chemical nature, even though it is a remarkable experiment. He conducted this experiment in pneumonia causing bacterium. The scientific name of pneumonia causing bacterium is Streptococcus pneumoniae. Direct question, you have to select third choice as our answer. Then moving to 20th question, the result of which of the following reaction experiments carried out by Avery, MacLeod and McCarthy on Streptococcus pneumoniae has proved conclusively that DNA is the genetic material. These three scientists proved or identified the chemical nature of transformation principle of Frederick Griffith. Frederick Griffith injected live R strain with heat kill strain to mouse, and the mouse were affected with pneumonia and are being killed. Then he collected the extract from such mouse and centrifuged. During centrifugation, they got an supernatant. Avery, MacLeod and McCarthy treated the supernatant, which contained lipid, proteins, RNA and DNA with their degrading enzymes respectively. When RNase is added, the RNA degraded, there also pneumonia caused. When protease is added to this supernatant, there also pneumonia. When lipase is added, there also the mouse afflicted with the pneumonia and killed. When they added DNA to this supernatant, the DNA are degraded. When DNA are absent in this extract, the rats will not affected with pneumonia, no pneumonia, and they survived. From this inference, they concluded that DNA is a genetic material in bacteria, or in the universal genetic material. So our answer to this question is option 4. So the next question, analyze the statements regarding the genetic material and select the correct option. The 4 statements are given about the genetic material. First one, genetic material has the ability to self-duplication, or they have the ability to replicate. It's a correct statement. Ability to replication is the ability of genetic material. Genetic material is very stable, correct? Does not change. This portion is wrong. The genetic material allows minor change, small changes called mutation, which are heritable to next generation also. The second statement is a wrong one. What about third one? Genetic material does not express itself. Genetic material does express. Genetic material have the ability to express the messages present in them. So the third statement is also a wrong one. What about fourth one? Genetic material stores information to control cellular function, cellular activities. It is a genetic material which controls the cellular activities. So fourth statement is a correct one. So here in the option, there are correct options are given. Correct options are 1 and 4. 1 and 4. That is choice 2 will be our answer to this question. Moving to next question, 22nd question. The scientist or scientists who conducted an experiment with P32 and S35 labeled phages for demonstrating that DNA is a genetic material. Here the experiment is conducted in T2 bacteriophages with radio labeled P32 and S35. This experiment was done to prove DNA is a genetic material in bacteriophages also. The remarkable experiment was done by Alfred Hershey and Martha Chase. This is a direct question and you can easily select the correct option. What about option 1? Watson and Crick proposed the double helix model of DNA. Frederick Griffith in the journey to searching genetic material. It is the first classic experiment by Frederick Griffith who discovered the transformation principle. Avery, MacLeod and McCarthy identified the chemical nature of that transforming principle. Yes, moving to next question. DNA is better genetic material than RNA because here DNA versus RNA. What is the better quality of DNA as genetic material? It is unstable and mutated at faster rate. This is the quality of RNA, not DNA. So it is wrong. It can directly code for synthesis of protein. RNA directly code for synthesis of protein. When DNA comes, it depends on RNA for expressing its character in the form of protein. So DNA have indirect access for expressing its message. So this second statement is also regarding RNA. The protein synthesizing machinery has evolved around it. It means RNA. The protein synthesis machinery consists of mRNA, rRNA and tRNA. Therefore, protein synthesis machinery revolves around RNA and not DNA. It is stable chemically and structurally. It is the character of DNA because RNA is not stable. Here the DNA is chemically and structurally stable one, double stranded. It mutate in a slower rate and it can store very well the genetic message also. So the fourth option will be answer to this question. Moving to next one, 24th one. Which statement is true about RNA? You have to select a correct statement regarding RNA. Stable and mutate at a slower rate. No, this is not the answer. It is a character of DNA. Primitive genetic material. We consider RNA as primitive genetic material and we have studied RNA world about RNA world also. So second option will be correct answer to this question. What about the other options? Indirect role in expression of character. This is a character of DNA. RNA has direct role, DNA has indirect role. Genetic material majority of organisms. This is also about DNA. DNA is the genetic material of majority organisms. RNA is genetic material of few organisms such as some viruses. So our answer is option 2. So we are going to next question, 25th one, statement type. Statement 1, semi-conservative mode of DNA replication was experimentally proved by Watson and Crick. It's a strong statement. We all know that semi-conservative mode of replication was experimentally proved by Michelson and Stoll. Yes. The statement 1 is a wrong one. Watson and Crick proposed semi-conservative. They proposed or they suggested the semi-conservative mode of DNA replication. Statement 2. Frederick Miescher identified that DNA is an acidic substance found in the nucleus of a cell. It is correct statement. Here, Miescher identified the first DNA and he named as nuclein to it. not DNA, nuclein. It is extracted from the nucleus of the pus cells. So our answer to this question is statement 1 is false, statement 2 is true. Fourth choice will be our answer to this question. Moving to next one, 26. Measles and stall grew Escherichia coli for many generations in the medium containing N15 nitrogen source. So initially the DNA in Escherichia coli will be having two heavy strands. This is also N15, this is also N15, two strands. Uh, then these bacteria are then allowed to grow in N14 source. NH4Cl. After 20 minutes, the kind of DNA strands will be. After 20 minutes, there will be semi-conservative mode of replication because there is normal nitrogen. Two new strands are formed. Here, 4 strands are there, 2 N15 and 2 N14. The question is after 40 minutes, again here semi-conservative mode of replication takes place. 8 strands will be formed. 8 strands. Of the 8 strands, 2 are N15 remaining. They are remained as 2 strands only, and 6 strands are N14. Of the 8 strands formed after 40 minutes, 2 retained as N15 and the 6 newly formed strands are N14. What is the answer? 2 heavy and 6 light strands will be there after 40 minutes. Choice 3 should be our answer. Moving to next question. Escherichia coli bacteria were grown in heavy nitrogen. And again, that type of question in new format. Heavy nitrogen, several generations, then it's transferred. Then here first it is heavy nitrogen, 2 strands. Both strands are incorporated with N15 isotope of nitrogen. So this is pure heavy DNA, pure heavy DNA. When it is treated with normal nitrogen for successive generations, due to semi-conservative mode of replication, the new strands added will be with normal nitrogen. You are growing the E. coli in normal nitrogen source in future, there will not be a pure heavy DNA molecule because we are giving normal nitrogen source. Therefore, our answer to this question will be 0. There is no possibility of forming pure heavy DNA molecules in succeeding generations because DNA replication is semi-conservative. Yes, moving to next question. 28th one. It is 5 statements are given. You have to correct answer. In Escherichia coli, the average rate of polymerization is approximately 2000 base pairs per second. Here, 4.6 into 10 raised to 6 base pairs are there in E. coli. The complete genome replicates with 18 minutes. The replication completes in 18 minutes. If we calculate the rate of reaction of that DNA polymerase enzyme, you can find that 2000 base pairs are added per second during polymerization. The statement is a correct one. In eukaryotes, replication of DNA occurs during G1 phase of cell cycle. It is a wrong statement. Not in G1 phase, it is in S phase or synthetic phase. In eukaryotes, replication of DNA occurs during the synthetic phase of cell cycle. It is a wrong statement as such. The replication of DNA in Escherichia coli initiates from a specific sequence called the origin of replication or ORI site. The statement is a correct one. DNA polymerase catalyzes polymerase only in 5' 3' direction. DNA polymerization takes place in 5' 3 direction. Correct statement. Leading to continuous synthesis on 3' 5' template. You know that in 3' 5' template there is continuous synthesis and the direction of synthesis in 5 prime 3 prime direction, and discontinuous in this one 5 prime 3 prime template. The statement is also a correct one. If you analyze the application for, you can understood very well this aspect. A failure in cell division after application of DNA results in the polyploidy. The statement is given in the textbook and it is a correct one. If the cell fails to cytokinesis, the result will be polyploidy. So analyze the options. Here only one statement is wrong, that is statement B. So our answer will be second choice. All statements except B are correct. Second choice will be answer to this 28th question. Moving to 29th question. The enzyme that is not involved in the process of DNA replication. Here DNA replication, the first enzyme topoisomerase, otherwise called gyrase. Gyrase. What is the role of gyrase? During unwinding of DNA for replication, the gyrase enzyme cuts and reseals the double-stranded parent strand to alleviate the tension due to unwinding. So, topoisomerase is involved in the process of DNA application, not the answer. DNA-dependent DNA polymerase, it is the main enzyme, main polymerase enzyme of DNA application. What of DNA-dependent RNA polymerase? It is also called primase. It is used for initiating the DNA replication by forming a RNA primer. Peptidyl transferase is not involved in the process of DNA replication. It is the enzyme involved in protein synthesis, which makes peptide bond between two amino acids. So our answer to this question or mismatched choice is peptidyl transferase. Moving to last question. In the replication of DNA, the small discontinuous segments are synthesized against 5 prime, 3 prime strand. The synthesis of these segments occur in. Let us imagine the replication fork. 3 prime, 5 prime. This is 5 prime, 3 prime. Here, small discontinuous segments are synthesized against 5 prime. This is a 5 prime, 3 prime strand. Here, the synthesis is discontinuous. Discontinuous. In 3 prime 5 prime template, there is continuous strand in the direction 5 prime 3 prime. Here in 5 prime 3 prime template, here the direction is also in 5 prime 3 prime. Direction will not change. But the type of synthesis is discontinuous. Here discontinuous. Discontinuous. But the direction is in 5 prime 3 prime. So you can strike out both 2 and 4 choices. You have to select answer from first and third choices. 5 prime 3 prime direction correct, joined by DNA gyrase. Not the answer. Here the enzyme is not DNA gyrase. 5 prime 3 prime direction and joined by the enzyme DNA ligase. Ligase is the correct joining enzyme here. So the third choice will be our answer to this last question. Hope you understood very well the 30 questions. Thank you very much.